And so I think that depends on the way that the cars are manufactured and developed and engineered. And so for a number of years, we had the construct where we built in shutoff switches within the CAR T construct. So if you do suspect, and we had a few patients where, there was a decision of, do we need to shut this down or not? And so it's always good to build in a safety mechanism into these products. So in our laboratory right now, we are validating a new assay on the Genexus platform, which enables us to be able to do both RNA and DNA sequencing at the same time. So I'm trained as a molecular pathologist, and since graduating from fellowship, this is the platform that I have really wanted to be able to interrogate both RNA and DNA at the same time within a day. And finally, we have instruments that can do that. Let me talk about, past standard of care for next generation sequencing and molecular assays. Typically, a next generation sequencing assay can be performed and from sample through reporting in, a stat case maybe 5 days, 3 days in some really high functioning reference laboratories and academic laboratories. But really about 5 days is the fastest you can get it. And typical is about 10 to 14 days when you get a molecular report with complete mutational profiling, and that's DNA or RNA, not combined. And so what we've been able to do on this new platform is actually do the sequencing at the same time with both types of nucleic acid. So now we can actually detect, say, fusion genes as well as expression at the same time with the variants and structural variations. The key about this platform is the RNA and DNA sequencing can occur within a 24-hour span. A day and maybe a day, a few hours, depending on which panel you're sequencing. So that is a game changer because you can actually pretty much report from sample to, sign out in about 3 days.